Welcome. My name's John Bagnulo. I'm excited to be with you today to talk about one of my favorite areas of human physiology, and that's the mitochondria. There's no aspect of cellular physiology that's at a greater interface with nutrition and our environment, and we're learning more and more about that every day. Today we're going to focus on mitochondrial health and all of the things that influence these tiny organelles and the amount of control they have over our health. It's a lot of information. At times it will maybe be a little technical, but I will do everything I can to answer your questions towards the end on that. Disclosure, I am the Director of Nutrition for Nutritional Medicinals. I've been in that role since 2014. My objectives for all of us today is to really understand how mitochondria, how they are damaged, and how that really starts to compromise our health. What are the foods and environmental exposures that do contribute to mitochondrial pathology or a loss of function? We'll take a look at the microbiome, and I would want everyone to understand how that can also play a role in mitochondrial health. And then at the end, really try to identify a pattern of eating in certain foods that are supportive of mitochondrial health. So that's really, those are my objectives. But I think before we go any further, it's really important to understand that mitochondria, again, if you don't remember, that biology course or that cellular physiology course from a long time ago, these tiny organelles are really where fatty acids and ketones are used to generate energy, to produce ATP. Those sources, those basically macronutrients derived from food or from body fat stores are shuttled into the mitochondria, and then through what we call oxidative phosphorylation reactions, they're in the electron transport chain. That's where we generate the largest amount of energy, or ATP. But because it's all based on oxidation, that there's going to be free radicals and reactive oxygen species as part of that. So they're very, very volatile reactions to some extent, and we have to make sure that the mitochondria are constructed in a way and have the defense system necessary to protect those organelles against these free radicals, and that's really, I think, fundamental to today's talk. Now, we know every single week at some research center across North America or the world, there's another paper being published that shows how a loss in mitochondrial health or a loss in the viable mitochondrial population for an individual, how that relates to a particular disease or pattern of diseases. And, we certainly have seen it within the cardiac, the field of cardiology and cardiac function. We know that a host of neurodegenerative diseases are tied to a loss in mitochondrial function. Of course, renal failure for quite some time now. We've seen the effects of that. And cancer is one that is more recent in terms of it being looked at as more of a chronic disease than simply a random mutation. Now, there obviously are mutations involved in the development of cancer, but we know that the role of the mitochondria in a loss of mitochondrial function is generally a common attribute across all forms of cancer. And then there's ataxia, sarcopenia, dementia, and other conditions where there is, again, an increasing amount of evidence that the mitochondrial function of an individual with one of these diseases is possibly one of the biggest determinants in the progression of that disease. So when we take a look at how some of these relationships might be founded, you can look at there's within the mitochondria, which you see here, that's the, this larger lower left corner of this slide. You're going to see that you have the Krebs cycle there. That's the TCA cycle that's going on, and derived from that, you have certain molecules, obviously acetyl-CoA being one of those molecules that's always being run through the Krebs cycle for for the production of ATP. But when the mitochondria are leaky and these acetyl-CoA molecules escape, they have the ability to drive, unfortunately, the histone unraveling, deacetylation, demethylation type processes, which we know are a big part of epigenetics and disease processes. And then if you don't have certain molecules being produced, some of these mitochondrial derived peptides that you see here on the left-hand side, if you don't have those being produced, then there's nothing to interfere with the processes of amyloid folding. And that's really where you get into things like many of your neurodegenerative diseases and Alzheimer's. So there are both really good things coming out of the mitochondria that are necessary to prevent diseases. And then there's some things that we don't want to escape the mitochondria that when they do really cause issues with gene expression. And that's where those reactive oxygen species and a leaky mitochondria contribute to some of those conditions. We know that in the area of sarcopenia, again, just to help everyone understand what sarcopenia is, it's a loss of functional muscle mass to where quality of life is compromised. We see that it's rampant now across North America. We have a large percentage of our population, not just over the age of 65, but actually over the age of 50 now, who have lost so much muscle mass that they're unable to, have some of the daily activities necessary for true independent living. It's really an epidemic, and we look at some of the factors involved, and obviously a lack of exercise, a lack of strength conditioning or weight-bearing exercises, just muscle is lost without that type of stimulus. But we also know that tied into all of that is a loss in mitochondrial function, and muscle has a very, very high mitochondrial density, especially slow twitch muscle fibers, which often serve as the foundation for most people's overall muscle mass. So is sarcopenia simply a function of inadequate exercise, not enough weight-bearing exercise and stimulating the muscle? Or is there something more to it where, yeah, those things are present, but also such a large percentage of our population has compromised mitochondrial health that that's contributing to this disease as well? And these are big questions, but we know that it plays a role to at least some extent in that. And as I mentioned just a moment ago, when it comes to renal disease, polycystic kidney diseases are definitely driven by mitochondrial dysfunction. Some of that has to do with the Warburg effect in the reliance on glycolysis. But kidneys form very, very high levels of cystic activity and lose function when there's a loss in that mitochondrial capacity there. So it's tied into most areas of medicine. Cancer, which I just mentioned earlier, is a big one. If you look at all of the common attributes of cancers and you look at how an over-reliance on glycolysis by those cells contributes to it, it's really clear. All those hallmark signs are exemplified by cells that have lost the ability to produce energy through oxidation through the mitochondria. And once a cell is relying solely on glycolysis, as is the case with cancer cells, you tend to have no negative feedback that would inhibit cellular growth. So you have uncontrolled cellular growth. There's no check and balance system, and that leads obviously to neoplasms of most types. So, again, it's really fascinating to think about cancer as more of a chronic disease, and the role of the mitochondria is functional to that. Here's a mitochondria, and you can look at the different ways where functionality is lost, right? You have just structural damage. There's different reasons why there's structural damage. One of the big ones is that the mitochondria in the mitochondrial membrane, just like with our cellular membrane, is largely comprised of lipids. And these lipids are fatty acids to a large extent. There's a lot of cholesterol there as well. We probably won't go down that road today in terms of the importance of cholesterol for maintaining this membrane integrity, but it is critical. It's irreplaceable, so you really want you don't want individuals to have large pools of available cholesterol to maintain healthy lipid membranes. That does prevent lipid peroxidation. Saturated fats and monounsaturated fats are the most, you'd say the most safe and less reactive types of fats to construct a mitochondrial membrane out of. And when those mitochondrial membranes are constructed of large amounts of polyunsaturated fats, especially omega-6 types, it really drives lipid peroxidation, and then you lose that membrane integrity, because once those lipids form these peroxide free radicals, it's in essence the membrane is being deteriorated quickly. You could use the analogy that's on fire because of the way those lipid peroxides cause chain reactions and basically cause a very, very high level of permeability. And then everything starts to leak out of the mitochondria. And that's why the earlier slide showed some of those intermediary metabolites leaking out of the mitochondria and then reaching other areas of the cell and causing gene expressions that we really don't want. So it's an increased porosity or permeability of the membrane. And that's because either there's too many free radicals, they overwhelm the antioxidant defense systems. Let's say this person has a selenium deficiency. If you don't have enough selenium, then you're not going to have enough of those endogenous antioxidants that the body produces. And that's what we see in people who have real severe selenium deficiencies. You call it white muscle disease, or a lot of different classic symptoms of that across all mammal species. But one of the things that happens with an extreme selenium deficiency is you have a loss of mitochondrial function, and it really compromises muscle function in the end. And we'll talk about carbonyl content and more about this, but again, two major mechanisms that are at play here when we have structural damage to the mitochondria and a loss in mitochondrial function. You have an excessive amount of omega-6 fatty acids being used that these mitochondrial membranes are comprised of, and then you don't have enough, an individual doesn't have enough antioxidants as part of their defense system to neutralize the free radicals that are part of mitochondrial metabolism. You need oxidation to take place. You just have to be able to neutralize some of those substances that are formed when it happens. And beyond this lipid peroxidation, which I've spent time talking about in the excessive reactive oxygen species, some other less familiar pathways that destroy the mitochondria are environmental toxins. There are pesticides that a lot of us are exposed to daily that can cause damage to the mitochondrial defense system, especially the superoxide dismutase enzyme. That's a huge part of our defense systems against free radicals. Some of those pesticides are again just on the average American's plate because of the types of fruits and vegetables that they're choosing. Then you have microbial infections within the gut, the microbiome. Blue-green algae is a great example of that. Sometimes we have exposure to certain forms of blue-green algae that might be part of an algae bloom on a lake, might be in some food that we've eaten, and then that will take a foothold in our microbiome, and it will produce a neurotoxin slowly over the course of our life. And by the time we're in our later years, it's taken its toll on the mitochondria of the brain. And then you have fructose metabolism, which I really spend a great deal of my research time looking at the various ways that fructose undermines human health, and this is a big one. So these are the five main pathways here where mitochondria are destroyed. The omega-6 content is a big one. Again, we have a very low ceiling of an ideal omega-6 to omega-3 ratio in all of our cellular membranes. That's mitochondria, as well as a phospholipid bilayer that wraps itself around all of our cells. That ceiling is somewhere around 3-to-1, maybe 4-to-1, but ranges, are estimated at somewhere between 30-to-1. That's that might be on the extreme end of things. And then they could be somewhere in the 20-to-1 or 18-to-1 omega-6 to omega-3 ratio. But even at that most conservative level, we're talking about levels that are 4 to 5 times higher. That ratio is 4 to 5 times higher than is physiologically optimal. So, our ancestors, due to the types of foods that served as a foundation for their overall pattern of eating, they had somewhere around 3-to-1. And now we're getting somewhere around, it might be 18-to-1, 20-to-1, some estimates are 30-to-1. When you have that type of increase in an omega-6 to omega-3 ratio, there are consequences That's largely inflammation, because omega-6 fats are used to drive inflammation. You end up with chronic inflammation. But another major area that this high omega-6 to omega-3 ratio is working against our health is how it undermines the mitochondrial membrane and how it really leads to excessive peroxide formation. And this is a great paper, by the way, by James O'Keefe and James D. Nicolantonio. I think their work, which is largely focused on omega-6s and omega-3s and how those turn certain disease patterns on, I think their work is really exceptional, and I encourage everyone that's listening today to look at some of the papers that they've published. It's really eye-opening. This is the pathway by which you could call these omega-6s as pro-inflammatory mediators. These, again, these might be in the form of arachidonic acid. This could be from excessive amounts of linoleic acid, which comes from some of those oils that we just highlighted on the previous slide. But when you're talking about things like corn oil, sunflower oil, safflower oil, even things like canola and soybean oil, there's a really large amount of omega-6 fatty acids inherent to those oils. And you want to minimize or avoid their use overall, because it contributes to the body's overall pool, and that pool of those fatty acids, that pool is largely maintained in our cell membranes and in our mitochondrial membranes. I mean, that's where those things end up. And so you're basically building those organelles out of highly flammable fatty acids. And we want to really, again, we want to construct those out of more saturated and monounsaturated fats, which are much more stable and are able to withstand the presence of these reactive oxygen species without forming free radicals or reactive oxygen species, and thereby leads to longer living mitochondria and much healthier mitochondria at any given age. These lipid peroxides, as I mentioned, again, those are driven, they're unavoidable. Okay, so you're going to have some of this lipid peroxidation, and I think really the important thing to understand here is that the scale is tipped on its end when you look at what has to happen in the mitochondria to produce ATP. You have to have oxygen. You have to have this as the primary method of energy production in the body. It's been said on many occasions, and I would completely agree with it, that one of the best ways to assess a person's all-cause mortality, meaning any population, any individual's risk of dying of chronic disease or dying at an earlier age than would be expected, is how much fat that they are able to burn at any given level of intensity, resting, exercise, regardless. The more fat we burn at any given state, then the more we're relying on our mitochondria for that energy production, and that's absolutely associated with a higher level of health and a lower risk for all-cause mortality. So we're going to have to generate ATP from the mitochondria, but we need to be able to do that without creating this chain reaction of lipid peroxidation. And so that's what we're talking about here when we're focused on these omega-6s and trying to minimize their presence in the diet. And when you look at cardiac tissue and you look at the ability for cardiac tissue to be more mitochondrial based and produce more energy, which we want. We want our hearts to use fat as a fuel. We want our heart muscle and our cardiac tissue to have very high functioning mitochondria. And omega-6s undermine that, and omega-3s really provide cardiac tissue with the type of mitochondrial function and mitochondrial output that in essence correlates with greater overall cardiovascular output. So the health of the heart is obviously tied to this, and a big part of that is the mitochondria within the heart. The peroxynitrites, they're probably the most damaging free radical that is formed. And again, it's to make matters worse, that's what the omega-6 fatty acids do produce. They produce, typically when they react with the most common free radicals inside the mitochondria, we're getting that as the highest concentration of reactive oxygen species. And those peroxynitrites, what they end up driving for as a catalytic conversion of pathology, it's the worst case scenario. So that's, again, that's the omega-6 component to this, which is significant when you look at the average American's diet and just the plethora of these industrial seed oils that are in most processed foods. You know, again, you have options, right? Olive oil, obviously, coconut oil, avocado oil. I mean, you have much better choices than the omega-6 dominant oil types. We'll take a look at that again at the end here. I mentioned environmental exposures, and one of those big ones that I just want to highlight here is the blue-green algae component, which is, again, a lot of people aren't familiar with this, but we have increasingly, it seems like every year, larger and larger amounts of water that people recreate on, both freshwater and saltwater, but especially freshwater, experiencing these cyanobacter or blue-green algae blooms. And some of those species of blue-green algae are really a high-risk exposure for things like Parkinson's and ALS. This is something that was first discovered at Dartmouth University back in the late 1990s when they, in their epidemiology work, discovered that there were these huge outbreaks of Parkinson's in and around all of these lakes in New England that were experiencing these blue-green algae. And it led us to understand more about the neurotoxins produced by that type of bacteria and how they can become more of a smoldering environmental exposure. Once those bacteria are ingested, and some of that ingestion can take place by just breathing the water spray that someone might be water skiing on or, you know, boating on. You ingest some of that blue-green algae spray, and it just kind of takes residence in the microbiome, and it produces this neurotoxin over the course of a person's life. So that's a big one. A lot of people are more familiar with some of the pesticides, rotenone being one of those, but there are others. There's a list now of common herbicides and pesticides that disrupt what I call that superoxide dismutase. It's a manganese superoxide dismutase antioxidant that's a big part of protecting the internal components of the mitochondria, and that is inhibited. The formation of that antioxidant is inhibited by rotenone. It's also inhibited by paraquat and chlorpyrifos. Now, chlorpyrifos is a really damaging pesticide. And again, the take-home message here is we don't want anybody, right? Kids, adults, we really don't want anybody to be exposed to these neurotoxins and these mitochondrial damaging pesticides. And they're a big part of agriculture, unfortunately, but we can avoid them by choosing organic versions of fruits and vegetables that carry the highest chemical burden. And I can talk about those in a little while, but you know, things like strawberries and grapes, you know, those are examples of pesticide-laden fruits that you really want to avoid unless you're going to choose organic versions. But this shows you how much that mitochondrial complex 1, that superoxide dismutase enzyme, is inhibited. You can see you've got, you know, significantly less here when there's exposure to those 3 pesticides alone. And when we look at the effects of that chlorpyrifos on the brain, which again, in brain function is tied to mitochondrial health, this organophosphate pesticide really affects children's brains. This is a really interesting paper, and it's 12 years old, but it's one that has stood the test of time, and a lot of pediatricians and epidemiologists in the area of pediatric health look at this and the subsequent studies that show how organophosphate pesticides really affect neurological health in children's mitochondria. And, you know, again, we have fructose as a big part of this conversation. And this is an area that I'm going to spend a little more time on than the others, because I think there's definitely a misunderstanding about fructose and the sources of fructose and how it can be part of a healthy diet. Never before in human history, I think it's important to say this at first. Never before in human history have humans eaten as much fructose as they are now. And it's not even a debate. I mean, you're talking about, by some estimates, you know, could be someone like Dr. Robert Lustig at the University of San Francisco, and he's not alone, but some estimates are that the average person's eating 100 times the amount of fructose today that our ancestors would have eaten as little as 500 or 1,000 years ago. And you know, the reason for that type of scenario, when you have that type of ratio or that type of extreme difference, is that you have people eating, you know, highly sweetened and sugar-containing foods. Sugar is about 50% fructose by weight, right? It's half glucose and it's half fructose. So fructose is what we call a monosaccharide. It's a one sugar unit. It's not combined with anything else. When you start linking fructose to fructose, you get other molecules, but fructose on its own or fructose combined to glucose, now that's sucrose, that's table sugar. But fructose is metabolized so differently than glucose. If you eat a white potato or you eat a sweet potato or you eat rice, the carbohydrates from those types of starchy foods are almost entirely made up of glucose. Now, fruit contains some fructose, depending on the type of fruit. Some fruit is very, very high in fructose, and some contains very little, but it's still fructose. So you have three different sources here. You have foods that contain starch and carbohydrates, but again, by and large, that's almost entirely glucose. There's sometimes very small amounts of fructose in things like a sweet potato, right, or a pepper, but those are small amounts. Then you have fruit, which contains some fructose. It still contains glucose, and the amount of fructose in any given fruit will vary significantly from things like mangoes, which have a very, very large amount, you know, pears, apples, grapes, and we'll have a slide that will show this in a moment. And then the third source of fructose would be in sweeteners. And most sweeteners, contain at least 50% of their sugar content in the form of fructose. So they're never good, but some, you know, like let's just say a entirely glucose-based sweetener, something like rice syrup, would not contain the fructose that something like a corn syrup, a high fructose corn syrup, obviously, or agave. Agave is the sweetener that contains the most fructose. It's like almost 90% fructose by weight. So here's the deal, though. When we metabolize fructose, there's a really important reaction that takes place where phosphorus has to be added. Phosphorus has to be added for fructose so that it can actually be used to generate energy. And when we add fructose to that, when we add phosphorus to that fructose molecule, okay, we at that point in time, we generate uric acid. That's an issue. And we also start to form a product called methyl glyoxal. And that's a really damaging molecule to human health. So these are glycation end products to some extent, but I consider fructose to be one of the most biologically non-compatible macronutrients in the human diet. We can handle about 25 grams of fructose per day. When we get to 50 grams, there's a consequence. The consequence might be the health of our mitochondria. It might be the health of our liver. It might be that we start to store more adipose tissue, more body fat. We might now start to produce more small, dense LDLs and be at a greater risk for heart disease. But make no mistake about it, when fructose consumption exceeds that and gets in the area of around 50 grams per day, there is a consequence to human health. And so this is one that I really like to put the spotlight on for everyone to understand. And a lot of that has to do with the way it's metabolized and how different that is than glucose. Now, methyl glyoxal, as I mentioned, is this byproduct of fructose metabolism. And there are endocrinologists now that are looking at this particular molecule, drawing it from a patient's blood and using that as possibly one of the best methods or biomarkers to track a diabetes patient's progress or ability to adhere to a better fit, a more diabetic type diet. So this methyl glyoxal, again, it's an advanced glycation end product to some extent. And it's one where we know it has multiple effects on our health, whether it's diabetes, whether it's on vascular health. We know that there's a loss in vascular elasticity. Our blood vessels become a little less elastic, a little more rigid and more susceptible to microvessel vascular diseases. And of course, methyl glyoxal given the talk we're having today, it damages mitochondrial health because it really ratchets up the level of oxidative stress inside mitochondria. So fructose, very different than glucose, and in many instances, they are discussed as though they're both natural types of carbohydrates or sugars and that they're both safe, and that's just not the case. So again, here's another paper that I encourage you to take a look at if you're interested in this, which is dicarbonyl stress. I mentioned that I would be discussing dicarbonyl proteins a little later on. Methylglyoxal is one of the most accurate and one of the most reflective biomarkers of biological aging. And so whether you're looking at mitochondrial dysfunction, you're looking at a loss of proteosis, and inflammation, as we talk about, just overall inflammation, we know that the amount of methylglyoxal that's present is a very accurate predictor of diseases that are a little further downstream. Obesity, diabetes, more upstream, but then you start getting into things like vascular dysfunction, or you get into neurodegeneration. Those are a little more downstream, but there's definitely a mechanism here by which methylglyoxal is contributing to the etiology of those diseases. So in summary, and this is again, this is really what I, want people to take home from this, is that dietary fructose, but not glucose. It's important. The carbohydrates that we get from things like rice, potato, carrots, even things like beets, which are sugary root vegetables, those are not implicated in this statement. I want to be really clear about that. Certain fruits are. We'll see it in a slide that it doesn't really matter if the fructose is coming from a sweetener or from certain fruits. The amount of fructose that someone is burdened with is going to be the number one factor here. But fructose, not glucose, impairs mitochondrial function, and that's and really results in a decreased ability to convert fatty acids to energy. And that's, this is the exact opposite, obviously, of what we want. So here are some fruits. You can see that you've got much better choices than others. Berries are always at the low end of the spectrum. You get up to grapes and mangoes, and things get high. I mean, I always say, like, lemons and limes have almost none. Cranberries, almost none. Things like raspberries and blackberries are a little bit better than blueberries. Blueberries are still good. When you get over, blueberries, you start to get in the area of apples and pears, and obviously you get down to dried fruit, things get out of hand. They get out of hand fast. So tangerines are phenomenal. You get less than 5 grams of fructose in a tangerine. That's fine. Most people handle 25 grams per day, 10 grams or less per meal. Those are safe ceilings that have been established through research on various mammalian models. But here's the thing that I want everyone to remember, is that the body responds significantly to high levels of fructose, regardless of where it's coming from. And you can see this paper was looked at apples, apple juice, and sweeteners, and compared them all, and they saw really similar effects. If people were eating large amounts of fructose from things like apples and apple juice, it resulted in higher levels of uric acid and all the things downstream from that. So the answer is to really choose fruits wisely and choose the appropriate amounts of fruits. And I'm not really here to malign fruits with a broad sweeping stroke and say that all fruit is bad and that people shouldn't eat fruit. But I think fruit needs to be looked at much more closely than it is. I mean, the message should be eat more vegetables and fruit, not eat more fruits and vegetables, because I think when a lot of clinicians hear that, they might not really appreciate the amount of fruit that their patients are eating and how it contributes to whether it's their diabetes or some of their other health issues. I think, choosing fruits wisely and keeping those to modest portions, maybe 2 or 3 maximum per day, is going to really pay off in the end for most populations. I'm sure there'll be questions about this in a little while, but fructose is a big part of this story, a huge part of the story, and that's why I wanted to give it the time. And again, that I'm not limiting this solely to the damage that fructose causes to mitochondria. We know that fructose also contributes to insulin resistance as much or more than any other type of carbohydrate. So, it's definitely better to have someone with diabetes eat some type of resistant starch-rich food, whether that's cold white rice or that's a baked potato. Those are going to be much better choices for someone with diabetes than is a as a bowl of some fruit that's very high in fructose. I mean, so this is stuff that most endocrinologists I think appreciate. So it's not limiting, or implicating fructose in only mitochondrial issues. We're talking about things like insulin resistance and obesity as well. Okay, so now let's try to tie some of this together and talk about how we can make, really a big difference in our patients' mitochondrial health, from what we've discussed so far. The core principles here are: use more monounsaturated fats and saturated fats as a foundation to cellular physiology. Getting more of the fat from things like coconut oil, butter, almonds, avocados, olive oil. That's always going to be the best way to build the most stable membranes in our phospholipid bilayer, our mitochondrial membrane, and that those polyunsaturated fats should be kept in balance. The omega-6 and omega-3 should be kept in much, much tighter balance, 3-to-1 max. 1-to-1 would be better as a therapeutic goal to try to reverse some inflammation and try to change some of that lipid composition of our membranes, try to get rid of some of those omega-6. We always say that, it's never too late to give yourself an oil change. There's studies, they go back to 1997 at Johns Hopkins where diabetes patients were given primarily olive oil as the only source of fat in their diet. And after 30 days with the use of an electron microscope, it was really clear. Those patients with diabetes that adopted a more olive oil based diet, they had an upwards of 40 to 45% change in their lipid composition after 30 days. So remember, it's a fluid situation when we talk about these phospholipid bilayers, these mitochondrial membranes. There's constant repair and replacement taking place with fatty acids. And so, it's always possible to give yourself that oil change that's going to produce a much, much higher level of stability in that tiny organelle. Try to find organic and chemical-free versions of your kind of go-to or staple vegetables and fruits. This is easy information to source now. There are a lot of different resources out there that will depict and will give you a really clear picture as to where the highest levels of pesticides, herbicides, and other agrochemicals are being found. So I don't need to go into that right now, but if you do some really basic homework here, you're going to find that there are much, much better versions than others. I talked about tangerines. I mean, tangerines are a phenomenal fruit. They're low in fructose, and it's not a fruit that's heavily sprayed, right? I mean, you've got choices like that. Same thing with sweet potatoes. Sweet potatoes are, a phenomenal source of carbohydrate, but it's the type of carbohydrate that's almost entirely glucose based. And sweet potatoes are really clean, even if they're conventionally grown. That type of work for your patients is going to really pay off for them and minimize their exposure to some of these. Avoid seafood from areas where blue-green algae blooms are common. I think we know now that there's certain times a year and there's certain areas of the Great Lakes, for instance, that we just don't want to eat. We don't want to eat fish from. There are also marine environments or seafood that we don't want to eat at times of the year when those bays are, really high in their blue-green algae and other algae contents. Shellfish are obviously another place that really harbors this. So you really want to get your seafood from cold water environments as much as possible. So that's an area where you're talking about minimizing environmental exposure. We've talked about carbohydrates, really eliminating fruit juice and fruit juice concentrates and sweeteners completely. Get those off of the table, off of the menu. Choose your fruits that are lower in fructose, like we've discussed. Really eat whole, minimally processed, unrefined plant-based diets. I mean, the word whole here is inherent to this. When you eat whole foods, you're always going to have typically an array of macronutrients that are more compatible with physiology. You're going to feed the microbiome in a much better way. You're going to have a lower glycemic burden. So that's always, I think, fundamental to this discussion. But choose whole, minimally processed plant-based diets that are going to provide not only the fiber and less sugar, but are also going to, give us the components to that antioxidant defense system that I spoke of. And again, summary here, when we take a look at the undamaged mitochondria at the top of this slide, you can see that there are attributes here, the alpha lipoic acid, the higher omega-3 content, those things are protective. We know that there are some different lifestyles and patterns of living, things like intermittent fasting, strength training, having different components to our lifestyle that might serve as mTOR inhibitors, intermittently at least. Those things are all going to drive mitochondria in the right direction, and might in some cases even drive the biogenesis or the synthesis of new mitochondria. And then you can see here all of those things that are typically associated with inflammation are going to drive the pathology of the mitochondria. And so, chronic inflammation is always public enemy number one. And when you have a diet that's generally supportive of healthy mitochondria, it's also going to be a way of eating, and it's going to have all of those macronutrients that are also supportive of anti-inflammatory processes. So that's a lot of information. I know some of it was technical. Again, it's a topic that I am always excited to talk about. I think it goes in a lot of different directions, from environmental exposures to some of the myths that persist around certain types of carbohydrates and certain types of fats that people have turned to in the last 40 years because they, are associated with a heart healthy diet, when I'm talking about some of these omega-6 rich industrial seed oils. So it's a topic I love to discuss. Now I look forward to answering any questions that you might have.
Great. Thank you so much, Dr. Bagnulo. While everybody's typing in their questions, and we have several so far, I did want to mention this webinar was approved for one continuing professional education unit for RDs and DTRs. You will get a separate email by the end of the day today that includes a link to our survey to give us feedback, and it also will include your certificate and a full list of references. So please be on the lookout for that separate email. So it won't generate after you close out today. It'll be in a separate email to the email you registered with. So with that being said, like I said, Dr. Bagnulo, we have a lot of questions here. I'm going to try and break them down by topics and start with some just general mitochondria questions. So the first one is, you mentioned that mitochondria can be improved with exercise. So can the number of mitochondria be improved with exercise, and does that include both strength training and cardiovascular?
Yeah, definitely. Typically the types of muscle we develop with strength training is more of a type 2 faster twitch muscle fiber. Again, I don't have time to get into some of the minutiae here of exercise physiology and kinesiology, but typically when we do strength training, we build muscle that has a little bit more of that fast twitch type 2 characteristic. So there's less mitochondria in that type of muscle to begin with, but there is some. So you're going to improve by building that type of muscle. You're going to improve your mitochondrial density, let's say, overall. But the cardiovascular exercise, that's really where you can make major improvements in mitochondrial density of any given muscle tissue. So absolutely. I also mentioned this process called biogenesis. We know that fasting, higher levels of activated protein kinase A, which happens with intermittent fasting, which also happens with a ketogenic diet, which in a way is mimicking fasting, that the presence of higher levels of activated protein kinase and sirtuin levels, those things are associated with the biogenesis or the production of new healthy mitochondria. And that's great. I mean, that's really, I think it's one of the more fascinating aspects of the benefits to fasting, intermittent fasting, and trying to place some type of caloric restriction, let's say, on the body. So the answer is yes. Strength training, some improvements. Cardiovascular exercise, or we would say more aerobic and endurance type exercise, which we call that slow SLD, right? Slow, long distance type exercise. Absolutely, you're going to improve mitochondrial density. And then non-exercise influencers would be intermittent fasting, and a ketogenic diet. All of those things have the ability because of their, when you fast or you're in a ketogenic diet, you're relying almost entirely on fat and ketone metabolism. And so once you really turn the emphasis on burning fat that you've ingested, or fat that through beta oxidation you're breaking down from the body, when those processes are turned up that much, there's a paramount, right, for having more mitochondria to metabolize fat. And so it's a stimulus to the body to get going, get more mitochondria available. So that's I know, a long-winded answer to that question.
No, that was great. Speaking of fasting, would you, when it comes to intermittent fasting, do you recommend that there be like in parentheses feast days with additional good carbs every so many days or weeks, or how do you feel like structuring fasting is ideal?
Yeah, that's one that's really, I think it just would be too individualized for me to give that kind of advice. I mean, there are people that, can do intermittent fasting almost indefinitely, and I think can make really some great progress to, like inhibiting the progression of their neurodegenerative disease or having major improvements in cognitive function. But then there are other people that, need a break from intermittent fasting, and certainly need a break from fasting, if it's any longer than that, intermittent type, because, there are certain components of our physiology that may suffer if we don't get even, modest amounts of carbohydrates from time to time. So that's a tough one. I think by and large, most people do really well with like time restricted eating. Let's say they eat all their meals within 8 hours and they have 16 hours of fasting, just a longer overnight period. I think by and large, that's really well tolerated, and most people do not need a break from that time restricted eating pattern. I think most people can do that almost indefinitely. Again, there are some exceptions, but by and large, it's well done. When you go beyond that and you start getting into, more restricted eating or longer fast, I think that's when people tend to need a window to step out of that.
Okay. Last question on fasting here. Is there any evidence that indicates that intermittent fasting damages thyroid hormone function?
No, I don't think we have evidence of time restricted eating or intermittent fasting. No, I think there is some evidence that with longer term fasting and with the case of a ketogenic diet, that there are certain populations that may be susceptible to seeing elevated TSH levels and some lower than expected thyroid activity.
Okay. We're going to pivot over to talking more about fats and the omega-3 to 6 ratio. So I know we had the one slide on the ratio of omega-3 to omega-6, but if you were to divide up the fat percentages, like if you're eating, say 100% of your fat intake, how would you divide it up into what percentage should come from saturated, mono, and polyunsaturated fats?
That's a great question. I think it's, I'll just try to give a general statement on that. I think around, somewhere around 50% of fat from saturated would be a good goal. I think, around, and then you've got, so you got another 50% of your fat you're going to account for. And I think that probably around 40% of that remainder would come from monounsaturated. So now I'm saying that 90% of fat is coming from saturated and monounsaturated fat. And that last 10% would be 5% and 5%. 5% omega-6s and 5% omega-3s. And that's for, in an ideal world, for a person, an adult living at a more temperate climate. I think that works really well. And someone might say, well, how do you know that? Well, if you take a look at mammalian tissue samples collected from a variety of species, humans included, that appears to be a physiological, optimal rate of fatty acid distribution. And with that, you get minimal susceptibility to this free radical damage in the mitochondria. You have a very, very stable membrane across all cell physiology. So that's how it would break down, Laura.
Great. And so speaking of that 5% from omega-3s, if you say you have a patient or yourself who doesn't eat any seafood and really doesn't get any sources of omega-3 through a whole foods diet, what is your take, or how do you guide people through like an omega-3 supplement decision?
Yeah, well, again, there's some huge variability here across human populations as to how much of the plant-based omega-3s, which we call alpha-linolenic acid, which comes predominantly from things like flaxseed oil, there is a huge variability in how much of that can be converted to DHA or EPA. In some estimates, it approaches 0% for one of those. That's for DHA. So it's really tricky to answer that. But I will say this: for women over the age of 40, you're probably only going to convert at most 20% of all the alpha-linolenic acid that you consume would be converted to DHA and EPA. For men, it's about 4%. That means that men need to eat a lot of flax oil to get the omega-3s that they need. We're just not very good at converting that to these long-chained, so they're elongated and highly polyunsaturated omega-3s. That's what has to happen for them to get to a place where they're physiologically something the body can use. We can't really use alpha-linolenic acid for preventing clot formation, for working against inflammatory processes, to have the right type of fatty acid shape and flexibility in a membrane. That alpha-linolenic acid needs to be modified by enzymes in the body. At certain ages, we're less effective at driving those reactions. Kids, pretty good at it. Women between the ages of like 10 and 40, really good at converting those. But outside of that, we need a lot more of that alpha-linolenic acid. So I would say, again, I'm not a big fan of using supplements. I don't know at the end of the day how much fish oil really helps. I think there's some important questions being asked about the efficacy of fish oil capsules. Do they even make a difference? It doesn't appear they do with things like all-cause mortality. But I think that, if you can, eating lower on the food chain, things like sardines and mackerel for people who are open to eating that, I think that's going to be your most effective approach to getting the omega-3s you need. Otherwise, you are going to be reliant on things like flaxseed oil in cold pressed versions of that oil, because it's going to be the easiest one to probably work into a diet. But I'm just not sure about fish oil. I think krill oil would probably be your best choice from that area, though.
Okay. And thinking more, a few people have actually asked this question. So for oils that are primarily monounsaturated fat, so your avocado oil, your extra virgin olive oil, I don't believe that chart listed those in terms of their omega-3 to 6 ratio. Do you happen to know how those stand up in terms of the ratio of those polys?
I do. They have low levels of poly overall. They have almost no omega-3s. None of those three oils, avocado, olive oil, or almond oil, none of those have significant amounts of omega-3s. But the good news is they do not have large amounts of omega-6s. Almonds and avocados have higher levels of omega-6s, but again, they're small relative to the overall monounsaturated content. That being said, you could easily overdo the omega-6s by using those oils with large amounts, or using them in larger quantities and very frequently, and not having the omega-3s present in the diet to balance that out. So when you get to the omega-3 content of oils, only flaxseed oil has the significant amount of omega-3s that we want without the concomitant omega-6s. I mean, people will point to canola oil as a good source of both, but you have too many omega-6s in canola oil for the small amount of omega-3s. So flaxseed oil is really the only oil that provides the omega-3s in the levels that we want.
Okay, great. Switching gears here, there are a lot of questions about the blue-green algae and such. So I'm going to ask a few questions, and then because they're all kind of linked here. So somebody asked, like, historically, there's documentation that people took ate blue-green algae for its health benefits. So one of the question is, can you talk a little bit more about how things have evolved with blue-green algae? They sell it as a supplement form, and just kind of go into that a little bit more.
Let me make sure I don't create widespread panic here with this. So spirulina is a member of the kind of blue-green. Look at things from a big kind of family of bacteria. The spirulina is categorized as a type of blue-green algae, but spirulina is not what is growing on these freshwater lakes when water temperatures get high and all of those phosphates from people's lawns leach into the lake. It's a very different type of blue-green algae. So I'm not talking about spirulina. I'm talking about the environmental blue-green algae that occurs with warm water. Spirulina grows on cold water in places like Klamath Lake and in the Pacific Northwest. But here we're talking about the type of algae blooms that occur in the summer months on freshwater lakes when there's a lot of organophosphates and fertilizers entering the water.
Okay, and so I'm assuming when they sell the blue-green algae as a supplement, that is the spirulina.
Yeah, you'd want to make sure they list the source and the subspecies.
Okay. Is there a lab test available for methyl glyoxal? And if so, what is the optimal lab range, if you happen to know that?
Yeah,
or it may be variable, but.
It's just not out there yet for practitioner use, but it's coming. I know researchers have put forth various levels that they associate with different levels of diabetes, in a patient's, ability to maintain better glycemic control. That I've seen methyl glyoxal numbers associated with that, but it's not available yet as a test that clinicians could order for their patients.
Okay. A pesticide question here. So in regards to, families who may come from lower socioeconomic statuses who may not be able to buy organic produce, what's the best way that they can treat their regular produce to get as much pesticide off? Like how much is removed from washing, or is there a vinegar mix, or what's your recommendation there?
No, unfortunately, it doesn't work like that. The vast majority of pesticides are ingrown into the flesh of a fruit or vegetable. So it's not like washing the vegetable or even peeling the vegetable is an effective way to mitigate that exposure. The Environmental Working Group, EWG, they do a really good job at 4 times a year taking all of the USDA data and some NIH data as well, and looking at food, mostly produce samples that are collected from various places across the United States, and looking at the measurable levels of different herbicides and pesticides. And then they put all of those fruits and vegetables into one of 3 categories: red light, yellow light, or green light. It's really easy to follow. I think, people in lower SES groups that can't afford the organic versions of some of these fruits and vegetables would benefit immensely from going to the environmental working groups, again, 4 times a year. So it's really up to date. Looking at that data, which is at their website, you can get these little printouts that you can put in your pocket whenever you go shopping, and just choosing from the green light foods, because those are the ones that have, non-detectable to very, very low levels of these pesticides and herbicides. That's the way to go. Choose things like sweet potatoes and broccoli over things like white potatoes and spinach. White potatoes and spinach are heavily sprayed, heavily treated. Sweet potatoes and broccoli, not very much. Same with grapefruits. Grapefruits are perfect. They're very inexpensive compared to other fruits when they're conventional. They have very low levels of fructose, and they're not sprayed, not significantly. So that would be my best advice in that situation.
Okay, and I think you said this, but just to refresh everyone, there's a few questions on what should be the limit of fructose consumption in a day? And then I think you also broke it down by meal. Can you go over that again?
Yeah, sure. 25 grams per day for adults. Might be less for kids, but Robert Lustig and some of his colleagues and other researchers that really look closely at the metabolic influences of fructose feel confident that 25 grams per day is a safe ceiling, and it's probably 10 grams per meal. Those would be your ceilings for adults. And I don't know what it would be for kids.
Okay, and is there any evidence or studies that have been done on athletes to say that maybe they can tolerate more fructose because they're more metabolically active, or has that not been accomplished yet?
I know the area of sports nutrition, really really looks closely at fructose for a variety of reasons, but I don't have any numbers that would suggest much higher levels. I mean, I think one theory that's been postulated is that athletes are going to be metabolizing fructose so much more quickly that it's going to leave them at lower levels of risk for things like diabetes and obesity. And I think that there may be some truth to that. The problem with fructose is, even if you're an athlete, as you might recall earlier in the presentation, I illustrated how fructose has to have a phosphorus molecule added to it. It has to be phosphorylated before it can be metabolized. So you're basically taking phosphorus in the liver and you're robbing Peter to pay Paul, in essence, and then you're forming uric acid with that. So I really question the efficacy or safety of athletes using more fructose in their diet. I think athletes are going to benefit much more from glucose than fructose.
Okay, great. I know we're right at 1 o'clock. Do you have time for 2 more questions, Dr. Bagnullo?
Yeah, sure.
Okay, the first one, this next one goes back to when we were talking about fat percentages and mono versus poly. Do you still feel like, even with somebody who has cardiovascular disease or high risk for cardiovascular disease, that the percentage of 50% saturated, 40% mono, and 5 poly still applies, or does that change?
I do think it can still work even for cardiovascular patient. I really think it depends on multiple factors. I mean, I would try to push the omega-3s up a little higher. I'd actually have a higher level of omega-3s to omega-6s in a person that I'm trying to, rehabilitate from cardiovascular disease. But saturated fat and monounsaturated fat, from all the evidence that I feel is out there, they're not really implicated in cardiovascular disease. It's much more about the other components and the other macronutrients, such as the carbohydrate quantity and qualities, the fructose. And I think that there's really good evidence if you start looking at Jim O'Keefe's papers, which I mentioned earlier, how I really like to follow his publications. He's got a great paper on omega-6 fatty acids in heart disease that I really encourage participants to look at. It would make you question the role of those in any cardiovascular patient's diet.
Great. And then our last question, I think this is a good one to end on, is, are there any significant efforts to stop the use of high fructose corn syrup in the food industry?
Yeah, absolutely. I mean, I know certain states have taken it on, individually, not federally, but I know like New York has taken it on, in foods that had a certain percentage of high fructose corn syrup as a major source of their calories were taxed accordingly. So there are efforts to do that. I don't think that they're far-reaching enough. I think there's a lot of momentum. Some of it's been able to carry through at that legislative level, but it's not at the federal level, unfortunately. But there are certainly some grassroots and more state-by-state efforts to prevent the high fructose sweetened beverages to being present in schools and places like that.
Awesome. Well, thank you so much, Dr. Bagnullo, for taking the time to answer all of those questions. I know we still had several more questions, y'all, so if your question didn't get answered, I really apologize for that. Thank you, everybody, for tuning in. It was an amazing, very, very well presented, complicated topic, but you really broke it down very easily. Thank you, Dr. Bagnullo.
Thank you so much, Laura, and thank you, everyone, for your interest in this. Take care.
Have a great day, everybody. Bye-bye.